thanks again for the invitation that I can give you some insights on or provide an overview on DNA RNA extraction technologies in general today. And in the second session, we will talk about more about fast DNA extraction and their use in point-of-care diagnostics. So, the history of nucleic acids and their discovery and research on them actually did not start in the last few decades, like most of you might know, I guess. But in fact, it was already 150 years ago that the first scientist discovered that there is what he called an highly acidic substance in cell nuclei. Back then, he investigated leukocytes from actual patient samples, and that was already in 1869. And according to these findings, that the new substance, which was previously unknown, was found in nuclei, and that it is highly acidic. That led to the fact that it was later named nucleic acids. The next big advancement was in the 1880s, so also quite some time ago, where Albrecht Kossel, a German scientist, determined the exact chemical composition of nucleic acids, which is the phosphate moiety, the sugar moiety, and the nucleobases. And that was also awarded, or he was also awarded a Nobel Prize in 1910 for this discovery. And I think the next big step, most of you know, that was when Watson and Crick deduced the double helix structure of the DNA. That is quite some interesting history, mainly based on the fact that they also got information from X-ray diffraction data from Rosalind Franklin, another very important person in that figure. And finally, that also led to another Nobel Prize in 1962. Yeah, and since the 1990s, roundabout, the technology was advanced enough that diagnostic assays based on detection of nucleic acid signatures became state-of-the-art in diagnostics, and they were then also called molecular diagnostics because we do not talk about protein detection, but actually this part that is really in the molecular structure of the cell. Right. So nucleic acid extraction is a very wide field, and I want to provide you first the core basic principles, how it always works. No matter how it looks, it always follows these principles. So first of all, you have to disrupt your cell. The cell shell needs to be lysed to release nucleic acids. And important also while doing this is to inactivate nucleases. That means DNases or RNases, depending on what you want to extract. You have to make sure that these enzymes do not degrade your DNA or RNA in the sample while you are preparing the nucleic acids. The third very important step is the nucleic acid purification itself. That means you want to separate the nucleic acids from other cell components like proteins, lipids, carbohydrates, but also smaller low molecular weight compounds called secondary metabolites, so that you end up with a very pure RNA or DNA. And while doing this step, usually you also have a concentration effect because mostly you bind your nucleic acids to certain solid surfaces, solid supports, and afterwards when you are done with the purification, you elute it with a smaller volume than you initially used for the cell extraction. So you have a certain concentration effect. That also, of course, increases your assay sensitivity. So whereas these extractions applied, of course, in research, in molecular biology or genetics, but also a lot, as I said, in molecular diagnostics, which can be infectiology, but also oncology or looking for inherited diseases. And I think also important fields like food safety testing or veterinary testing, but also forensics. All those rely heavily on the extraction of nucleic acids. So how did the extraction look like 150 years ago when Friedrich Miescher did it the first time? So you see the scheme, it always was the same, it always will be the same. And in the 1870s, Miescher, as I said, worked on leukocytes. And what he did in his early protocols was to wash them several times with warm alcohol, which provided a certain lysis of the cells. Then he deactivated the nucleases and also broke down, of course, proteins of the cell itself by a very long protease digestion. And afterwards, he washed these liquids several times with ether and alcohol to extract proteins and lipids. And these alcohol precipitated DNA, he then later on dissolved again in a mild alkaline solution. And here you see we are through all the 4 steps that I introduced to you. And that was the protocol he used back then. So, of course, in the last decades, there were a lot of additional ways and modes being revealed and discovered and developed by scientists how to perform the 4 individual steps of the DNA and RNA extraction. And if you look at the cell lysis, it depends on the sample specimen how harsh you have to do the lysis. For example, viruses are very easy to extract. Similar animal and human cells is also not such a big deal because the cells disintegrate very easily. But if you want to extract DNA from fungi or for gram-positive bacteria, you really have to do more, have to take more effort because those cells are very hard and it takes some effort to break the cell walls. And how is that done? So you have basically in total 3 different possibilities. First of it is the mechanical treatments. So you can ground your tissue or cells. You can use steel balls, for example, for plant tissue to really grind off the cell wall. For gram-positive bacteria, for example, you would use glass or ceramic beads and do a strong bead beating. You can also use a French press or ultrasonication is also possible. So all these are more or less mechanical treatments. And there are other ways, are chemical treatments, which rely on the use of detergents, of alkaline conditions, the addition of enzymes that break up some cell wall components. You can also use a simple osmotic shock. You put your cells into water, basically. Of course, it doesn't work for all cell types, but for example, for red blood cells. But also you can just add alcohols or organic solvents. This would also provide some cell disruption. And the most simple one is to apply heat to the sample. So thermal treatment, usually you would heat the sample above 90 degrees. That is also very efficient for breaking up cells. And if it comes to the nuclease inactivation, people usually use either denaturating conditions like certain concentration of SDS or high concentration of chaotropic salts, or you can also inactivate enzymes by denaturating them through heat application. Another possibility is to apply enzymes, especially proteases, to inactivate RNases and DNases. So if you look at the second half of this workflow, we are at the nucleic acid purification. The degree of purification depends a little bit on how robust your assay is, what type of detection assay you use for detection. So there's really a wide variety of how to do the purification. And I try to summarize them a little bit and make certain categories. So one of them is solvent extraction of the impurities, and we talk about proteins and lipids as impurities here. You can do this with organic solvents such as ethyl ether or phenol. It's very common. Usually you use additives like chloroform or cresol. Another possibility is to precipitate proteins, especially. You would do that by addition of SDS and later on addition of high concentration of salt ions like potassium or sodium. And then the proteins precipitate, and you can centrifuge them or filter them away. And yeah, you got rid of this part of the impurities. Another possibility is to scavenge the impurities by something like size exclusion chromatography, where you run your sample through chromatography, and it's basically a gel filtration process where all small molecules and compounds like salts and nucleotides and short oligonucleotides would basically interact with your column, and the nucleic acids would just flush through, and by that you can separate them. And meanwhile, not used procedure anymore is the ultracentrifugation based on a cesium chloride gradient. Was very popular in the mid of last century, where you can easily or very nicely separate your nucleic acids according to density in this gradient, and then later on prepare it and use it for your downstream applications. Another very important aspect of this purification is the actual precipitation and binding of the nucleic acids themselves, which is basically used by all the standard purification methods. And for that, again, you have different possibilities how to do this. First of all, you have to provide precipitating conditions. You do this by addition of high salt, high chromotropic salt concentrations. So we talk about guanidinium or sodium perchloride, or especially well working is guanidinium thiocyanate. It's a very strong chromotropic salt. Another way of providing these precipitation conditions is the addition of ethanol or isopropanol, means alcohols, or high concentration of sodium chloride and PEG, polyethylene glycol. This depends a little bit what solid support you want to use for binding the nucleic acids, and there you also have certain possibilities. Like one would be glass or diatomase resins as a solid support. Very similar are silica membranes or silica coated magnetic beads. They are combined or they are used if you provide the precipitation through chromotropic salts and ethanol, for example. And but there are also other supports like carboxylated resins or carboxylated magnetic beads, which can be used if you precipitate your nucleic acids through the addition of sodium chloride and PEG. And another rather new way of adsorbing the DNA and RNA is the use of cellulose filter papers, or there are also cellulose magnetic beads available from commercial suppliers, which also work together with the chromotropic precipitation mechanism. And one other way, not so often used, but still also in use by some commercial suppliers, is the electrostatic binding of nucleic acids to anion exchange resins. There you, by a simple pH shift, you can switch the resin to be binding or not binding to the RNA and DNA. And the nucleic acid concentration is basically inherent to this mechanism because, I mean, it's not always required, of course, because some assays do not need a very high sensitivity or the nucleic acid is abundant anyways. But how it is usually done is just the step after the precipitation. After you have bound the nucleic acids to the solid support, washed away all the impurities, then you would elute the nucleic acids again with a certain elution buffer or rehydration from an ethanol precipitation into a smaller volume than you initially had as a sample volume. So really the most important, or let's say most often used way of doing the purification is the chaotropic salts combined with addition of alcohols, and then usually the nucleic acids are bound or attached to the silica membrane or silica beads. Now, if you look through, let's say, the last 70 years of nucleic acid research, you see, I just draw it up here and pointed out which method was developed in which time. What you basically see that's very interesting is in the last 20 years, we didn't have any more really new inventions in terms of purification technologies. Everything was basically kind of invented up to the year 2000. But what you see from there on, there is a certain trend towards less purification. And why is that? The one driver for this is point-of-care diagnostics. I will explain this a little bit or go more into detail in the next webinar session. So this is because the point-of-care diagnostics provides more rapid results to the patient, or another driver for this point-of-care diagnostics, more profit to the physician. And of course, you don't need really a central lab, a big lab facilities anymore, which is more important for emerging countries where this huge central lab infrastructure is not as developed as, for example, in Germany or in the US. Second driver is indeed the technology. In the last 20 years, we saw that instruments like PCR cyclers or instruments for isothermal amplification became smaller and smaller, much more energy efficient, and also the amplification became shorter and shorter. And the third driver is the application itself. The PCR master mixes. If you look at the development of PCR master mixes, you see that the vendors advertise more and more that their master mixes are resistant against certain inhibitors, like blood stains and stuff. So that means the amplification chemistry is nowadays much more robust. If you talk about simple PCR or real-time PCR assay, than it has been 20 years ago. And so, of course, the purity of the DNA and RNA doesn't need to be as pure anymore as it was in the early days of nucleic acid extraction. So depending on your application, you can really work with not so pure nucleic acids. That means the removal of all these impurities like proteins, lipids, carbohydrates doesn't need to be 100% anymore. Usually it's good enough if it's less than this ultra pure state. And that's why you have a trend towards less purification, meaning faster extraction and not such a high purification, or also not such a high effort for the purification step of the extraction process. So what's the difference then between this fast extraction and the standard extraction as I explained to you? Here you see again the 4 steps. Basically, you can't change anything with the cell lysis and the nucleus inactivation. That needs to be the same. You need to get rid of, or you have to break up the cells. You need to get rid of the DNA's activity or RNA's activity. But where you can change something is the second half, the purification and also the concentration. And if you talk about this fast extraction or direct extraction, then we would not talk about an actual nucleic acid purification, but rather about an inhibitor removal. It's just a difference in point of view. And how do you do this inhibitor removal? There are certain measures. I have listed it here. You can, for example, do centrifugation. If you do it before lysis, you get rid of all the inhibitors in your sample matrix. Before you start with the extraction, you would get rid of them and remove them out of the equation so that they can't disturb your assay anymore. Another way of doing centrifugation is after the cell lysis. When you have your cell debris or precipitated proteins, you would centrifuge them down and use the supernatant, which contains the nucleic acids for your assay. Another possibility is simple gel filtration that would remove salts or low molecular weight compounds, let's say everything below 10 kilodalton molecular weight. There are possibilities to add polyvinyl pyrrolidone, PVP, which is known to bind phenolic compounds. It's more important if you, for example, extract nucleic acids from plants. Another way published is the addition of Helix 100, which can bind divalent cations, which usually are important for DNA's activity. And another way is the addition of so-called reverse purification beads, which basically bind cell debris and proteins and carbohydrates. And last but not least, if the assay allows, if the assay doesn't need a super high sensitivity, then you can even just dilute your crude extracted nucleic acids. Of course, you dilute the concentration of the nucleic acids itself, but the more important step here is to also lower the concentration of the impurities. And if you get below the threshold, which your assay can still stand, then this simple method works as well. And yeah, as I said, the nucleic acid concentration is usually not done by the direct extraction, but if you would do a centrifugation before lysis, where you basically pellet your cells of interest, then you would even have a nucleic acid concentration effect. Right, and that was my last slide for the first session. I thank you very much. I have listed here all the references I worked with, and yeah, thank you for your attention.